Billions of years ago, long before the first cell emerged, Earth was a chemical playground. Life as we know it hadn't begun, but the ingredients were swirling in ancient seas. Among them were sugars. Not the kind you stir into tea, but simple 5-carbon molecules, fighting for a role in the chemistry of life. One of these was ribose. And strangely enough, this humble sugar would go on to become the backbone of RNA, the molecule many scientists believe kickstarted life itself. But why ribose? Why not one of the many other sugars with the same chemical formula? A new study from Scripps Research may have the answer. The researchers recreated early Earth-like conditions and tested how 4 similar sugars, including ribose, reacted with a compound called DAP. Short for diamidophosphate. This molecule is thought to have existed before enzymes, and it can donate phosphate groups, an essential step in building RNA. In modern cells, this job is done by enzymes. But billions of years ago, DAP may have helped sugars and phosphates join hands, without life, without biology. What the team found was striking. Ribose reacted faster, more cleanly, and more selectively than its chemical cousins. Not only did ribose form the ideal structure, it did so with a rare precision. Here's why that matters. For RNA to exist, ribose must fold into a very specific shape, a 5-membered ring called the furanose form, made of 4 carbon atoms and 1 oxygen atom. This is the exact structure seen in real RNA molecules. The other sugars, like arabinose, xylose, and lixose, formed messy mixtures, some 5-membered rings, some 6. Worse, they often got stuck halfway through the reaction. But ribose not only completed the reaction, it did it with elegance. In a mixture of sugars, DAP consistently favored ribose, attaching phosphate groups almost exclusively to it. And the resulting molecules were perfectly shaped to bond with nitrogen bases, the next crucial step toward forming real nucleotides. This doesn't mean ribose was destined to become life's chosen sugar. But it does suggest that its unique chemistry gave it a head start in the primordial race. And perhaps, just perhaps, this chemical edge nudged evolution of life in a particular direction, one that led to RNA, to cells, and eventually, to us. The next big question? Whether ribose can do the same thing inside protocells, tiny bubbles that might have been life's first containers. If it can, scientists might finally be able to watch life's earliest steps unfold, one reaction at a time.